talking about evidence.
It's not focused.
It's blurry.
There you go.
There, I fixed it. You're welcome.
We're talking for evidence for evolution. Like I said, it's more important for the sound to work than anything else. All right, so then what I want help with, and let's get sigils here as part of the mix. What I want help with is deciding from what fields of science, and this is also indicated right there on the board, so you can look at it. But from what fields of science do we have information about evolution as a thing that actually happens?
You're missing
Right.
one.
So let's see. Natalie Rios, are you here? Or are you a senior? She's not here? Are you here?
She's right there.
Okay, so give me a field of science that gives evidence to support evolution. A type of science.
Biology?
Okay, obviously. An area of biology that helps us support evolution actually happens.
No?
What do you mean by history?
All the
Okay, fields of science that support evolution. So in your reading, we learn about different types of evidence that exist for evolution, right? So supposedly, like a field of science, I'll give an example. Well, maybe somebody else can help. Yes? Paleontology, you see? Paleontology. You know what paleontology is? Natalie Rios?
No.
You don't know what paleontology is? You're gonna learn about it today. All right. So we got paleontology. That's a good one. So we don't know what a word is like this. We can actually kind of tie this. Like, ever heard about paleo? Ever before? The paleo diet? Ever heard about that? How about Paleolithic in history class? No? Not that either? Anybody can help me here then. Let's see. Paleo. What does paleo mean? You know what it is? Yes. No, nothing to do with natural. It's because paleo diet, it's because it means rock. But paleo diet is because it's the supposedly like the diet of their ancestors of a long time ago, which is the natural way for humans to eat, so forth. Paleontology is the study of rocks, but more specifically, the fossilization of life found in rocks. So then that's a good thing over here. And of course, from that, we have fossil. All right? Fossils. Now, of course, fossils, how do fossils provide evidence that life evolved? Lots of different ways. So let me see. Kevin. Kevin out here? Yes, give me one way in which a fossil supports the life. Remember, what is evolution? Maria. Define it for us. Evolution. Okay, you can't use the word, okay, they change. They study how life changes over time. So if we're looking for evidence of that, we're looking for evidence that life has been changing. So, Kevin, how does a fossil record show us that?
We can study the bones, the structure of the bones.
Bone structure, all right, so, sure. We can do bone structure. Or in general, actually, let's just call it anatomical structure. We can tell that from the fossils. But what about it? What can we tell? What's happened to the anatomical structures? What can we see in fossils?
Like there's like vestigial structures and stuff. But
That is, okay, let's bring in that field of biology then there. Let's talk about anatomy then. How, and then that connects with the fossil record in a way, right? So let's put that in there. Because now, what kinds of anatomical evidence show up? Now, she mentioned one over here. The first one was vestigial. All right, so somebody tell me what a vestigial structure is. Let's see. Stephanie Kiros, are you here today?
Right.
Okay, what is a vestigial structure? Did you read this chapter yet? No? Okay, so you got to play catch up, all right? I know you're playing catch up. Cheryl, yes. No idea? It's okay if you're not sure. Now is the time not to be sure. On the FRQ, you got to be sure. You can make a mistake if you think. What does it sound like, vestigial? What does that sound like? Any idea? Okay, I'll give an example, and maybe from that you can know what it is. The appendix is a vestigial structure. So what is that? A structure that has no use or less use than it used to have for an ancestral organism, right? But we have it because our ancestors had it, and we're still in the process of like getting rid of it. But now that's a tricky thing because that's only going to happen if the pressure to get rid of it continues, right? So there's a lot of examples of vestigial structures. In
But-
biology, we should do a lot of examples.
But-
So like we talk about appendix, although there's some research that suggests the appendix may have immunofunction. For example, it may be an area that holds E. coli and it helps prevent infection from other nocive bacteria. But it's mostly a little area that was back a long time ago when we came from organisms that were less, not less evolved, because that's a different, I think that concept, whoever said less evolved, don't use that, because evolution is not purposeful. It's not trying to actually go somewhere. So when we think of a human as more evolved than something else, that's actually not an accurate way of thinking about how evolution is. Evolution is not trying to get better, make things better. And you could even argue that better depends on the situation. And that we've talked about this before. Who's better, the elephant or the cockroach? It depends on what the environment is, and they're not even competing. It doesn't even make sense to think about it that way, right? So don't use that. But anyway, what I was trying to say is the appendix may have had a function associated with longer intestinal tract, and some herbivores and also some carnivores, they have these little tracts between the large intestine and the small intestine. So for extra digestion and absorption of food. But since our digestive system is longer and more efficient, and the fact that we cook our food, right? It makes it less necessary for us to have that. So then there's pressure to lose it. Why is there pressure to lose it? Because if you have something that you spend energy on, right, that to make, but you're not using it, then somebody else that's not spending that energy to make it is going to have an advantage over you, right? Or you can have infection and it died in there, but someone that doesn't have it or have a smaller one will have a less chance of infection. And that is what sets the pressure to lose it. For example, I just read an article about they found out this fish that lived in a cave, and it actually lost a large part of its brain and a lot of all its eyesight. And they have evidence that happened over with fossil evidence because it was more of it didn't need to see, it didn't need to process visual information. So it was actually better for its survival to not invest the energy on a useless thing like that. So that's a good example of that. Now, some more hands went up for vestigial structure, so I guess you guys know more. Yes.
Isn't like example of it, like the molars are no longer
Mold,
Wisdom
teeth, wisdom teeth. Good. Yes. Tailbones of. Actually, we even have a coccyx, right? Something like that. Yes.
Like the pelvis on the wheel.
The pelvic bone in a whale. All those are good examples. Yes. Make sure those are good. Lego givers, make sure they're getting Legos. Yes.
Can I get a Lego?
Sorry?
Goosebumps?
Why do you think it's vestigial? I never heard about that.
Because they said that goosebumps, we used to get them because our old ancestors used to have feathers, so then we have like goosebumps that where we used to have.
But I thought that's used for like.
Okay, so she's saying our old ancestors has feathers. The problem we didn't do a history of life yet, but the problem I have with that is that no, I don't think it's any mammalian. We don't came from a lion that's feathers, so that's not accurate.
No, but
A bird line and a mammal line split a long time ago, and feathers evolved in a bird branch. Actually, birds come from a branch. Like think about the reptilians, the early reptilians, right? Those early reptilians, the Therapsidia, which is what animals came from, we split from that a long time ago. And then this branch, you have things like turtles, alligators, dinosaurs, birds. Birds is a cousin of dinosaurs, which are cousins of alligators, which are cousin of things like turtles, right? Which is then a cousin of the therapsids, which is where it came from. And then feathers are here. So that doesn't make sense. So I'm not saying it's not a vestigial structure, but that reason, that can't be that reason. So look it up. Teach me why it's vestigial. Yes. Hair. Oh, we do have hair. We still have hair.
eyebrows.
Well, I know that dogs, they kind of use like their hair stands up as a way of some dogs and some animals have like this hair that stands up in the way of demonstrating either aggression or fear. You know, so maybe it's something to do with that.
Can I look at a map?
So sure, yes.
I don't know if this counts, but I heard that the pinky toe, that we have it, but we don't really need
That's
it.
sure. There's a lot of other things. Although I heard it's used for balance. There's also muscles that are used to move the ears. That we still have the muscles, even
I could
though
do that.
most people can't access them or use them.
I got him.
But the muscle's still there. Alright. There's or actual this is not actually useful.
Your eyelashes too.
Right? Although it may have a kind of sexual selection thing. It may just be for purely aesthetic purposes nowadays. You know what I'm trying to say? But there's evidence that they're actually getting shorter because it's actually not useful. So, anyways, the vestigial structure. But this is such a ginormous evidence for evolution, and I like that we brought that up early in the class, because why would a designed creature have useless features? So that doesn't make sense, right? So why would a designed creature have a useless features? It's almost as if you know how Windows, even Windows 10, in its programming, it has remnants of the programming of Windows a long time ago. Now they have revamped it a lot since Windows 7. Windows 8 and 10 were very different. But still, in the mechanics of Windows 10, there's still age-old programming, right? And it's almost like Windows is what it is because of what it's been. You follow what I'm trying to say? And so it inherits the history of what it was been. And in the code, this is going to be super important soon, the code of the Windows programming inherits the code of its ancestors. And a lot of those features are actually not even useful anymore. There's a networking from Windows NT features, which are often most Windows developers can activate that if they need to. But for the most people, it's like vestigial. It's not useful, but it's still part of what you install when you install the software. And that's why it wastes a lot of space and stuff like that. So people like Macs because they just kind of like, they don't have supposedly they don't have useless stuff in there that you're not going to use. But the point is vestigial. It's very difficult to explain how a design thing would have the vestigial thing. It's almost as if the architect would have been like, oh, I kind of use this feature on this animal. On this one, I'm just going to leave a little piece of it just because. You know, just because it's beautiful. You know? Now you start arguing that, and now you start arguing, well, we are not here to argue the reasons our God is doing their thing. You get another type of anatomical structure, Ines.
I follow the Stephen.
All right, let's move on though, because I have a lot of things to do. Yes.
Like the bone structure and different positions that like how they stand.
What do you mean?
Like for example, I read that like horses when they first started, they were very different, but they were
Okay.
four-legged.
So what you're talking about is transition in the bone structure. All right. So we call this, and we actually call this transition fossils. With transition fossils, we can actually see the history of anatomical changes. All right. So I'm going to put like HS. You can see the history of anatomical structural changes through transition fossils. You can actually see a picture of the evolutionary process. Let me ask you a question. Is there anyone alive that has seen the sequoia trees grow in Yosemite Park, California? Have you seen an entire sequoia tree grow? No, because it's 1,000 years old. So there's no one that has seen a sequoia tree grow. Is there any doubt in anybody's mind that the sequoia tree started with a seedling, and then it sprouted, and it grew more and more, and eventually it's that thing? For some reason, people have no trouble accepting that. That you start from a seedling and you go to this enormous tree. Even though nobody has seen it. But why not? Because if you walk through the forest, you see the seeds. You see the seedlings. You see the early stages. Because throughout the forest, there's trees growing at different stages. So you can picture from that a transition. Right? The fossil record is no different. If you see so many transition fossils, and the humans are included on this, there's a lot of transition fossils for humans. Not everything. That's another thing that's interesting. I'll put a little parenthesis here. There are missing links all over the place. And by that we mean the fossil record is not perfect. It only conserves fossils that are conserved, are the ones which were either accidentally lucky to be conserved because of the conditions that the person died into, or the animal died into. Usually they're not conserved in places like, which have decomposition rates which are really high. Unless there was a sort of flood or something like that. But you're more likely to conserve something in a forest than you are in a desert, where life is so rare to begin with. So desert life forms are very rarely represented in the fossil record. Aquatic life forms are represented more than anything else, right? Because they died and sediment fall covers them. It just makes sense. Now, hard shell or hard pieces that conserve a lot more than others. So organisms with hard shells, like shellfish, right, that conserve a lot more than say a flimsy little mammal, where only its bones perhaps get conserved. So you see, that the fossil record conservation is not perfect. And because of that, we have a picture full of gaps. right? But that doesn't mean that we can't tell something from the story from the gaps we have. When we go to a crime scene and we see the crime that just happened, we can piece the crime together using science, despite the fact that nobody has actually been there to witness it. Even without a witness, somebody can go to jail. Because we piece the different pieces. Even though the whole picture is missing, we get enough pieces to tell the story of what happened, right? If that process was not trustworthy, why are we putting people in jail? The same philosophy that allows us to do that kind of science allows us to do this kind of thing with physician fossils, all right? But I was actually talking about a type of anatomical structure that is used as evidence of evolution. Yes. Homologous structure. That's another one that I was looking for. So homologous. And again, these are things that we can see that in the fossil record throughout the transitions that we have. All right. So, by the way, let me throw another thing that fossils help over here. We also have things called index fossils that help a lot. Index fossils are fossils when we don't know, right? Oh, by the way, another thing that comes from paleontology, I will also add here geology. And geology is a cousin of paleontology in a way because it's the rock. The rocks are preserved, right? Life is preserved in the geological record. But in the study of geology, we have things like radiocarbon dating. All right. But more importantly, all right, the uranium to lead dating series. People make the mistake of saying, oh, carbon dating is not accurate beyond 20,000 years. Therefore, it's stupid to use that as evidence for evolution. You're probably right. Unless you're talking about recent evolution, that makes no sense. You're talking about things that are evolving since two ice ages or so. Because anything beyond that, the whole way, we're not supposed to know how carbon dating actually works, but it's just supposed to know it's used as evidence, but it's actually an exclusion thing. The details of the methods are beyond the scope of the course, right? So nobody's going to ask you about how carbon dating works and how half-lives are calculated. You're not going to have that question. But you need to know that they do that. But basically, life forms are, while you're breathing, you're taking in carbon dioxide, the plants are taking in carbon dioxide, and then you eat plants. So you take the carbon dioxide from the plants. But because of the way the nitrogen is struck on the atmosphere by cosmic rays from the sun, some of the nitrogen in the atmosphere undergoes a radioactive decay process that makes carbon-14. And so long as the sun is around, that's going to happen so long as nitrogen is around. So carbon-14 is made in the atmosphere. It's like a carbon-14 factory. And that allows us to have a count of a percentage of the air that's carbon-14, right? But if the air is X percentage is carbon-14, then the plants X percent is carbon-14, because X percent of the carbon that the plants are going to pick up are that, right? And then, therefore, X percent of the food web carbon is going to be carbon-14, right? Now, if a plant lived today, it could sit on the plant for thousands of years, so that in the sequoia trees, some of the carbon-14 on them is decaying on them as they live. Are you with me? Because some of the carbon-14 they captured 1,000 years ago is already decaying. But the half-life of carbon-14 is 5,000 years. So even for the largest of trees, the majority of the carbon-14 that they consumed will eventually pass to the next item of the food chain. You follow what I'm trying to say? But my point is interesting is that should be that X rate of carbon-14 on you because you're consuming it. But when you die, you stop consuming carbon-14, and then what happens is that your carcass, over time, all of the carbon-14 in it decays to carbon-12. And by the rate of what should be a carbon-14 in there, because of the atmosphere composition, and what it actually is, the difference between it, you can use to time it, right? But the problem is that there's so little carbon-14 to begin with. It's such a small percentage of the carbon in you, that's carbon-14, that by the time 3 or 4 half-lives go by, the amount is so small that it's not precise enough to use for dating anymore. So no scientist will use carbon-14 dating for anything more than 25,000 years. So if you debunk or find something that, if a scientist were to find something that debunks carbon-14 dating as a way to evidence for evolution, you'd be like, no problem. That's not what scientists use anyways. Again, I love the idiocy of people that fight without understanding what evolution is. So when they go like, oh, evolution is wrong because carbon-14 dating doesn't work. It's like you don't even know what you're talking about. We don't use carbon-14 for evolutionary dating. In fact, evolution, geological history, we're talking about eons. And for that, carbon-14 doesn't make sense. On the other hand, uranium lab dating is very precise. When volcanoes erupt, a little bit of uranium is always in the lava, right? Because it comes from the core of the earth. So the mantle of the earth, it has an X amount of uranium. But once that rock becomes a rock, no more new uranium is going to go into that rock. But since this uranium was radioactive, over time, the uranium decays through a long series of decay processes, which eventually ends in lead. But that whole entire series takes about 8 billion years to occur. So that means we can actually have a clock that ticks very slowly, that allows us to see the passage of time. You can date very ancient rocks in this manner. Right? If you find a rock that has no uranium in it, it's probably very, very ancient. You know what I'm saying? And so uranium lead dating allows us to date rocks really well. But sometimes we actually date through the fossils. If we know there's a fossil in this layer that only occurred, that the animal went extinct 65 million years ago. So you find a Tyrannosaurus rex in the layer of rock. That rock has to be at least 65 million years old, right? And that's how we know, sometimes we know the ages of rocks based on the living things we find in it. Because if we know, now it may seem like a backwards way of thinking, because you just say, you date the rocks, all right? You date the life forms from the rocks for the age of the rock. But sometimes you date the rock from the life. So people use like, oh, see, circular logic, error in logic in evolutionary research. Not really, because we're not doing that for the same rocks. Somewhere we use the rocks to date the fossil. Somewhere else, because we already dated that one, we see the same fossil. It's like, oh, the rock is the same date. You see what I'm trying to say? It's not like we're using the same rock to date the specimen and then the specimen to date the rock. Are you following what I'm trying to say? And also, all of this stuff is corroborated by a lot of different types of evidence. So it's not just you have dating, rock layer dating, and all other things it's a lot of things put together, but there's more. From geology, you can look at erosion rates. All right, you can look at rock formation rates. From geology, you can also look at rock composition. You can see what the rocks are made of, and that can help you tell the story of life. We'll talk more about that in a second. All right? Homologous structure. We're talking about this. That is a structure that is what? Similar, but it's in two different types of life, right? So the function is the same, right? The structure is the same. So like the bones of our hands versus the bones of whales' hands and things like that. We'll see, if time permitting, I'll show you more pictures of all of this in the board in a second. So homologous structures, bone structures that show similarity, all right, for example, can be
What about
ancestral.
embryology?
All right, analogous structures is different. Analogous structures is also similar structure. Now, there's a way to differentiate both of them. With homologous structures, you have the same ancestor, and then there was a split in the evolution process, and that's why these two things have that same characteristic, because they share the common ancestor. So when us and the whale share similar bone structures, it's because us and the whale share a common ancestor. How do you know the difference? If the fossil of the common ancestor, right, shows that structure, and then the fossils of the people they're comparing show that structure, and most likely it's because they share that structure, and that's the homologous structure. So the homologous structure will be present in some form, even though it's modified, because remember, what's happened here since, these organisms have different evolution. So they started from the same point, but they evolved different because of different environments, different, genetic drift, all the different 5 things that make things evolve. So because of that, they will have differences. But yet, the things which are similar between them are most likely because of common ancestry. Analogous structures is different. Analogous structures, that you have 2 things that started from a different point. And for the most part, they are still different. They are still different. But there may be one feature that they both gain, which is similar. Now, that feature is not present in the ancestor. I'm throwing a little plus sign there, right? So it's just a logic diagram, right? So logic diagram is to represent that. So you have 2 organisms that are mostly different, but yet they are still mostly different, but they gain something that the other one also gained somewhat. Now, that's because of similar pressure, similar evolutionary pressure, right? So, for example, the wings of bats, the wings of birds, the wings of reptiles, right? Because reptiles had wings. You mean a reptile with wings? Pterodactyl, right? How about a mammal with wings? A bat. How about a invertebrate with wings? A fly, right? Now, and a bird has wings. So different groups of life have wings. What's the deal with wings? Because the pressure to fly is similar. But here is how you tell them apart. Aside from actually having the common ancestor to look at, if the common ancestor has it, then it's homologous structure. The other way to tell them apart is that homologous structures have a high degree of congruency. What that means is that they are very, very similar. Or analogous structures, they may have a similar purpose, but the structure that fulfills that purpose has a large degree of variety. So when you compare the wings of a bat and the wings of a bird and the wings of a fly, they're very different wings. Yes, they're all wings, and in a sense they all have wings, right? But the structure of the wings does not indicate homology because doesn't have a high degree of anatomical congruency. You know what I'm trying to say? So usually you can tell that apart. It gets difficult with things like internal temperature control. All right? So like, for example, birds and mammals have the same kind of internal temperature control mechanisms down to the way the brain works, some of the chemicals which are involved in controlling the process, right? And the metabolism that is involved in actually maintaining your temperature high. But it looks as if the common ancestor of birds and mammals did not share that characteristic, and therefore it's not a homologous structure, it's an analogous structure. But the problem is that how can such complex, down to chemicals that regulate it, have evolved in two separate branches of the tree of life? We'll talk more about that when we talk about phylogeny and classification, but that creates a problem, because then we think about, hold on a second. If birds and mammals share a very high degree of congruency in some sort of feature, doesn't that mean that perhaps they share a common ancestor more recently than we thought of, right? Because other organisms which are their ancestors do not have the characteristics in common, right? That raises a question, and that's why we think of trees of life as hypotheses that need to be confirmed with a variety of evidence, right? We'll talk more about that in the weeks that come. But in general, it's very easy to tell them apart because even though they are similar structures, these ones don't have a high degree of congruent similarity of anatomical structures. But this is such a big thing too. You can see the evolutionary evidence for that. Now, people argue this, it's just the same designer making the same thing in 7 different places. But this one is hard to argue with that designer thing, because then you have to accept your designer to be flawed and purposely put things which have no purpose, just to waste energy on your things. There's another type of anatomical structure that's mosaic. Mosaic structures are anatomical structures which have evolved for one purpose, but are not being used for others, or are also sometimes described as structures that are combinations of many other structures. So when you look at, for example, the flagellum of a bacteria, it seems as if it would be almost impossible for something so complicated to have evolved, because the thing is that any one given part you take out, it won't work as a flagellum anymore. That's a common argument used against evolution. It's the idea of the irreducibly complex system, like the eye. You take anything away from the eye stops working, and therefore it's useless. It couldn't have evolved because the whole thing needs to be there for the eye to work. Well, there's a lot of errors of logic with that kind of thinking. First of all, who says that a broken eye or an eye was missing a piece is useless? So if you take away, for example, the cones, that's fine. You still have the rods. You can still see something, right? Without the cones, you don't see color, right? But with the without the rods, you don't see color. But with the cones, you can see contrast. Or you can see which part is dark, which part is lighter. And so you can still go towards the light. So there could still be a benefit as long as you can have a benefit, even if it's a small little increase in probability of survival. So you know that the microevolution simulation that you have to do, right? If you do a simulation, do a 2,000 generations, and make the selection feature 100% chance of survival for one gene, and the other gene you just put 0.98. The difference in survival is so small, right? You would think nothing would happen. But even though it's only 0.2 different, over 2,000 generations, you will see the gene doing the same thing it does. It will just take longer, right? But the rate at which evolution happens, it takes longer. But the selective pressure is still there, so over long periods of time, the gene will be selected against, because there's a slight advantage over the other one. So all you need is like slight advantage. You don't need to have perfect eyes for the eyes to be advantageous. Ask anybody who's blind if they wouldn't rather at least see the contour of stuff. So when they're crossing the street, they can tell there's a bus there, right? So there's a misconception that a system has to be perfect in order for it to be advantageous, right? The flagellum is a good example. There's a lot of in-between steps that could have been beneficial for the bacteria to get to the point that it is today, the bacterial flagellum. But my point is, if you look at bacterial flagellum, there's evidence that it actually evolved from several different types of proteins. So each protein evolved for a different reason, but then they were kind of combined by accident to form a flagellum. at some point, and then more features were added little by little. But the point is, it's a mosaic structure in the sense that it didn't evolve all at once. Each part of it evolved for a different reason. The motor protein that spins the flagellum was actually the same kind of protein that's used to make energy in another part of the membrane. So it's just a cousin of that. So think of it as that it's like, for example, a cell phone. It's a mosaic structure, or cell phones of today, because we invented cameras, we invented cell phones, we invented computers, right? And the cell phones that exist today are mosaics of those things put together. So the cell phones that exist today weren't developed to do everything they do. What they were developed to do was to put all those things together, right? Evolution is kind of like that. Sometimes you have things that evolve for a reason, combining with other things, or being modified to use for another reason. Your hand is a mosaic structure. Because this evolved for walking, right? It was a foot at some point. But now it's been modified for grasping. But what I love about mosaic structures is something else that's evidence for evolution that has to do with all of this too. I think I would fit this in the kind of idea of paleontology too, which is the idea of evolutionary constraint. I'll put that as a thing here. Evolutionary constraints. That's what these anatomical structures such as vestigial structures show. Mosaic structure shows that. Homologous structure shows that. It shows that the way you are today depends on the history your kind came through. So you have features because your ancestors had those features. For example, your hand looks like this because the ancestors we came from had bone structures that made our hands was modified, right? From another hand, from another limb, right, or other tool. But that tool, because it's modified, it will never be the best grasping tool there is. It won't be, because it will always have the constraint of having been in the past a walking tool. And because of that, we have limits to how it can bend and turn and stuff. Are you with me? Because this wasn't designed for grasping. I love that about mosaic structures. It's another great evidence against the design. Because if I was designing a grasping tool, I could do a better job. You know what I'm trying to say? Because, seriously. And I'm not even like a god. But the thing is that this thing here has a problem. It's a problem that comes from the ancestral constraints. You can think of it as vestigial structures that limit what we can do. Right? We will never be the best graspers until we redesign this hand. Because this hand was not built for grasping from the start. So all we did is we modified. It's kind of like getting the Empire State Building and say, now I want it to look like a pyramid. Right? So you could do it. But the fact that it was always a long building will always affect your design of changing it to something else. You know what I'm trying to say? So it was set up, that's why cell phones are never going to be the best cameras. You want a good camera, you buy a dedicated camera. Because there's constraints in the design of the cell phone from the history of what it has to do, or where it came from. You know what I'm trying to say? Until we were willing to give up some of those things, we can't improve on the cell phone enough. All right. So this is a lot of evidence here so far. Now, there's a lot of also, and it fits with this, the rock composition. There's another kind of thing which we'll call chemical evidence of evolution. Chemical evidence of evolution. All right? So for chemical evidence of evolution, we can talk about biochemistry. All right? Or we can talk about this kind of thing, like actual physical chemistry. And I connect that to the rock composition because it has to do with that. So for example, when I find oxygen in rocks older than 2 billion years, but not in rocks that are older than 3 billion years, that tells us that at some point there was an oxygen revolution that happened. And why does that oxygen revolution happen? It corresponds to the fossil records indication of the origin of photosynthetic life, right? And so we have these fossils called Stromatolites, which show us that, I assume you have read the origin and history of life, so you should know what I'm talking about. So we have all fossils named Stromatolites, which show large colonies of photosynthetic bacteria. And those fossils only date back to about 2.3 billion years, and before that, you don't have that. So the evidence that we have from paleontology is corroborated by physical chemistry evidence in the rocks that show the lack of oxygen in ancient rocks, right? So you could get that evidence if you needed to corroborate that evolution happened and that caused that change in the order of life. Biochemistry, looking at DNA protein sequences, we can then compare and do those kinds of things. For example, why do we have DNA on us that's homologous to bacterial DNA? All right? Because we as bacteria do the same functions. We have proteins which are similar in our membranes. We have proteins which are similar in our, for example, cell respiration. The proteins that do glycolysis, the enzymes that break down glucose are in us and are in a lot of other organisms. So that kind of degree of homological similarity, right, down to DNA level. Now, here's why this evidence is better than this evidence. In here, you can just count how many features these animals share in common. But in here, you can actually quantify, give a score to the homology, which is the next lab 3 we're going to do towards the end of this month, right? On lab 3, we do an analysis of DNA sequences using a software that actually quantifies the amount of difference that exists between the genes. And from that, you can actually talk about how many base pairs are different. And since the DNA is a long sequence, you can give a score that has a lot of meaning, a mathematical value for the amount of difference between two organisms. And then you can rank organisms or group them together that way and build better trees of life because you have this kind of data. So the cool thing about molecular data, molecular biology, right, in biochemistry, let's put molecular biology in there too, molecular biology, is that, same kind of idea, is that you can quantify it at a level that was never be able to quantifiable before. But the DNA record also has vestigial structures. What do I mean by that? Let's see if the next person does. Sundip, do you know what I mean? Where's Sundip? Do you know what I mean by vestigial structures on the DNA? Sorry. What does that mean? A vestigial structure in the DNA. We talk that what? What is a vestigial structure in anatomy? It was an anatomical structure that we no longer use, but that we have because what? Ancestors had something that was useful for that, right? So how would that be? How would that look on a DNA code? It would be a sequence that what? It's just there. It has no purpose for us, but that is similar to a sequence that what? Our ancestors had. So for example, we have DNA in us that we call the pseudogenes. Okay? Pseudogenes. So pseudogenes are vestigial DNA structures. Literally, they're genes which are broken, shattered, never readable or expressible, but they in those sequences, they are highly congruent to the sequences of other genes from other life forms where they may have had a function. So we have biological comparison genes. Think about it. Vestigial structures are kind of like homologous structures. We can even put a little dash mark here, right? They're homologous in a sense, right? The pelvic bone of the whale is homologous to our pelvic bone. That's why we know it's vestigial in the whale, right? That's this homology, so to speak, in DNA code, but these codes are broken, and yet there. So you can think of them as an archaeological evidence of our evolution. Because again, why would we have DNA code just like a bacterial DNA, right? Except not useful, broken, sitting in our DNA code, right? You could think of our DNA as a library full of old broken books with missing pieces that have no more useful information except that page is very similar to the page of the library of the bacteria, where that book also may be associated with a feature that a bacteria does. So that's interesting, right? Why do we have pages of their library in our library, right? Because our library was copied from the library that was copied from the library that was copied from the library where we both came from. You know what I'm trying to say? And that's why we have that. We also have analogous structures in the DNA code. Sometimes, for example, the genes for the enzymes that control the temperature regulation in humans and in birds. They're analogous structures because the pressure was so similar that the protein structure is very similar. And so the genetic sequence is similar by an accident because of pressure selecting for that accident, right? So, and then of course, mosaic structures, full of DNA sequences which are actually combinations of different genes. The immunoglobulin gene, genes that make your immune system. We'll learn about that later in the year. These genes, they operate through a combination of several different genes put together. And that's why you can make a variety of different antibodies. Because what it is that you have this data bank of possibilities. And the way you do it an antibody is by piecing together different pieces of this data bank. So it's a perfect example of mosaic evolution at the genetic level, right? Genes that evolve for one reason combining to do other things, right? And so that's the new kind of evidence for evolution. And the thing is that when you can quantify and analyze in a mathematical way, it really becomes hard to deny it. Along with that, we have another kind of awesome piece of evidence. It's called molecular clocking. Molecular clocking. And this is like, what do you know? This is it. This is the nail in the coffin of anybody that is against evolution, right? Because here's the interesting thing about molecular clocking. You can get the mitochondrial DNA. So the thing about mitochondria is that your sperm of a guy never goes inside the egg of the female, right? Because the female's egg, only the DNA of the sperm goes inside, which means all the mitochondria you got it from mom, right? So mitochondria has DNA though, because it's a living thing, technically. So all the DNA you got from the mitochondrial DNA, it's coming from the maternal line. So if your dad has a mitochondrial DNA disease, a mutation that's causing a mitochondrial problem, you can't inherit that because it comes from your mom. So it's a non-Mendelian pattern of inheritance. You inherit the genes from the maternal line only because it comes from the mitochondria, right? Now that's interesting. But the point is, if you could do that, you can actually trace down the DNA of the mitochondria down to figure out your family's history. So for example, if I get your mitochondrial DNA and I do an analysis of that, I extract the DNA, I put it in the computer, use a machine to make multiple copies of it, and then I put a computer to analyze it and read it. And I get that code and it combined with, and I test it against a data bank on the software that I'm going to show you guys, right? So you test it against a data bank of mitochondrial DNAs from lots of humans from all over the world. You can actually see what percentage of your DNA is from, say, a certain place or a certain culture. That's why people say, I found out I was Irish, right? Because you do DNA analysis and you find out that you actually have DNA similar to that culture or to that group, right? Not culture. Culture is social. I mean, to that ethnic group, right? To that race, so to speak. And that's actually very interesting. Now, if you do that kind of analysis, you can actually, because here's how this works. It's one more level than that. If you know how often mutations happen just because of accident, the rate at which mutations happen, and we know this because we know the rate at which we fail to correct mistakes. Now, mutations can accelerate during times where solar radiation is more intense or chemical exposure to chemicals is more intense. But on average, the rate at which mutations occur because of errors in the DNA copy process is pretty constant because it depends on the biochemical machinery. So we know how often the DNA replication process makes mistakes which are not caught. So we have what, think about it, is the mutation speed. You know what I'm trying to say? Now, if you know the mutation distance, you divide that by the mutation speed, you get what is called mutation time, right? It's like physics, right? Velocity is distance divided by time. But if you have time, if you have velocity and distance, you can solve for time just by messing with the equation, right? Think about it this way. I know how far I am from some other organism because I compare my genes and the genes of the gorilla, and I find that we're 98.1% the same. So that means we're 1.9% different. But I also know the size of my genome, so I know what 1.9% difference means. I know how many base pair differences that actually means. In this software, I can actually spill this kind of data. So if I put a gene in there, and I say it's 99.9% the same, there's 5 base pair differences. But I also know the rate at which mutations happen. So if I know there's 5 base pair differences, right, and mutations happens at a certain rate, you can actually use that to calculate how long ago you split from that group. Now, this is insane because this is the most interesting part. This is new stuff, right? This is from the 21st century, right? So we used to do this, we started this after the Human Genome Project, and then with all kinds of organisms. Now, the cool thing about this is that when we do that, and we say, for example, analyze the gene of a fish, and then analyze that against the gene of us, the hemoglobin gene, for example, the gene that carries oxygen. And you compare that to the gene of the fish and our gene, right? Now, hemoglobin is such an important gene that it can't mutate very fast because if it mutates, you die, right? But so it's an important gene, so it mutates slowly. That's the thing about the molecular clocking too. There's different types of clocks. An important gene mutates slowly because it's very, very strong pressure to keep it the way it is. Does that make sense for you? A gene that's not important, like a pseudogene, for example, that's useless. It's just sitting there in the library. if there's a mistake, is anybody going to notice it? No. So those mutate very fast. Are you following what I'm trying to say? So genes which are less important, less crucial, mutate very fast. So those are good genes if you want to look at things which are recent changes, because the clock is ticking fast. So I'll give you an example. If I want to time how long this takes to fall to the table from here, I need a stopwatch that has decimals of time. So a stopwatch that's ticking fast. You know what I'm trying to say? But if I need to know how long this takes to fall from the moon, probably a wall clock with seconds will be enough. Hell, I only need minutes for that, or even hours. You know what I'm trying to say? So the precision that you need depends on how quickly the thing is happening. Are you with me? So if I want to see the difference between you and me, I need a clock that ticks very fast, because that's not too long ago. That's what? We must share a common ancestor. I don't know, maybe 10 generations back, at least. You have ancestors from Spain, right?
I guess.
Probably at some point. Sorry about the wrong assumption, but most of us are Hispanic here. So if you have an ancestor from Spain at some point, I must have had an ancestor from Spain at some point, because I'm also Hispanic, right? Kind of, right? Actually, I'm Portuguese. But Portuguese and Spain came from the same ancestor at some point, which means, I don't know, it can't be more than 1,000 years. Somewhere along the lines, we share an ancestor, right? But 1,000 years in geological time, that's a tick, that's quick. So this is why mitochondrial DNA is awesome for that. Because mitochondria mutate very quickly, because they're bacteria, so they don't have DNA control mechanisms to repair the way we do. So that means mutations in the mitochondrial DNA gather quickly. It's a fast clicking clock. But if I wanted to compare me and a fish, I need a crucial gene, because that changes very slowly. I found what I'm trying to say? But anyways, when you calculate us versus the fish hemoglobin, and you use molecular clocking, and you find out the number of base pair differences that exist in the hemoglobin gene, and you times that to the mutation rate that we have, the average mutation rate of eukaryotic genes, you get a number like 300 million years old. 300 million years have gone by because of according to this molecular clocking. That is exactly how long the fossil record indicates that we should have Precambrian time. Now, it's two completely separate times of the guy that was doing molecular clocking wasn't trying to get the same numbers, right? And that's what's interesting about it. Two completely separate branches of science giving you the same time for that fossil, for that connection between us and the fish. Because we split from the fish millions of years ago. Actually, it's not that like that. The fish that exists today has been evolving too, right? But the fish family that exists today and the mammal family that exists today, when do they share a common ancestor? All the way back to Precambrian time, right? And so that means that's 350 million years ago. And so it matches molecular clocking information, right? And that's super, super interesting. So that's, for me, it's like a nail in the coffin of anybody that's against evolution, because you can actually be like, okay, me and her, how long ago has it been that we shared a family? Get our mitochondria, get my mitochondria, compare. See how many base pair differences there is in our DNA. Let's say we got 5. Let's say the mutation rate is 50 years. That's 250 years. We do some genealogy research and find out we have an ancestor. Guess what? 250 years ago. That happens. We've done that kind of research. And I think it's mind-boggling for me that we can use that, right? So that's cool. That's pervasive evidence that we can actually trust that evolution is a thing, because the science behind it is corroborated by a vast amount of research. Just to make sure we didn't forget anything else. We're talking about physical evidence, geological evidence. Oh, geographical evidence. How could we forget geographical evidence? All right, so I'll fit it in here. Geography. What do I mean by geographical evidence? Let me see. Samantha Valdez, are you here? Okay. Do you have any idea what I mean by geographical evidence that things evolve? By looking at places?
Yeah, like there's fossils in one area in the world, also certain fossils in other places.
Right? Which shows that at some point, these fossils kind of must have been what? Together. That's one way of looking at it. What is another way of looking at places as evidence for evolution? Why are, for example, all marsupials, a majority of marsupials, why are they in Australia?
Because they have similar pressure. And
It's
of the
probably because
environment.
that's where the marsupial type evolved. I found what I'm trying to say? And this pressure is very similar there, right? So it stayed that way. Now, why is it that different, for example, the beaks of the finches? This is geographical evidence. It shows how it's matched for its environment, right? That is actually one of the original pieces of evidence that Darwin presented to show that there is corroboration between the life forms and their environment across space and time. And that's kind of why we talk about geographical evidence as evidence for evolution. And there's another one that I forgot here that's kind of a cousin of anatomy. It's morphological embryology. Embryology. Now, I love this kind of evidence because, again, what the hell was the designer thinking when they made you a tail when you were 2 days old? Or 2 weeks old. At 2 weeks of age, you have a tail, like a lizard, right? So that is interesting. And at 3 weeks old, if you get an embryo that's 3 weeks old from a human, and a 3-year-old chicken embryo, unless you're an embryologist, you won't be able to tell the difference between them. And that's because the genes which are activated in both types are the same. So they look the same. As human genes, peculiar human genes, primate genes, start activating, we start looking and developing differently. But we have the same genetic code for a lot of it. More, like, we are 70% like a fish. So that means at the beginning, yes, we have gills in our embryo because those genes are on. But then as other genes turn on and others turn off, in the sequential development sequence, you actually change that. You know what I'm trying to say? So embryology is an awesome evidence for evolution because, again, it shows you that it is undeniable that we have evolved because we actually have these genes. We actually even look as our ancestors look, right, in their embryological development, right? So we talked about all of this already for the most part. Vestigial structures, biochemical genetic similarities, in particular DNA and protein sequences. And the last one is mathematical evidence. All right? So math. I know. When I see the time running out, I usually do that. So mathematical evidence. So mathematical evidence, I show several examples of this. You can quantify with molecular clocking. You can count base pair differences to see similarity, right? So this kind of ties with this other evidence from here. And we also have things like Heidi-Weinberg. You can actually predict the number of genes that are in a population and how it's going to happen in the next generation based on your analysis of the quantity of genes in a population. So graphical analysis, which we're going to do in lab 3. Basically, this last piece of 1.A.4 is all about lab 3. Graphical analysis and mathematical analysis of DNA pair differences allows us to prove basically that evolution happens.
They always made that dangerous for the third period.
For third period, that's right. It's okay. So, anyways, we didn't actually, for that time that I wasted with the bit in the beginning, it took away the 15 minutes that I needed to talk about essay writing strategies. But it's not a loss since we're not going to write essays today, so to speak. But any questions about evidence for evolution before we start? Yes, go ahead.
Penguin would be homologous.
To what?
The wings of a penguin because they have a common ancestry with birds.
Right, they are birds. They are birds, and they have wing-looking things, right?
But wouldn't that be vestigial? Wouldn't that be vestigial because they don't really use their wings? Not homologous?
They have modified it to do something else. It's mosaic, right? Because they use it as a fin almost, right? Which is analogous to the fins of fish. You know what I'm trying to say?
Yeah.
All right. One thing I wanted to show you before we leave. All right. So please switch the camera back to the main board. You can actually access this. It's on the videos and web tutorials folder for evolution. By the way, I'm going to throw this out there. Anybody who's curious or interested about evolutionary theory and its research and the data behind it, okay? Everything we're going to talk about here in class is going to be sufficient for you to understand the kind of things I'm talking about, and plus the reading you guys have to do. So remember, all we do here is integrate. I don't have time to cover all of this in detail. I just kind of do an exposition, right? But what I'm trying to say is that if you go to this videos and web tutorials folder, okay, you will find in there this thing. Let me show you. Come on. There's in the videos and web tutorials folder for evolutionary theory. So if you go to unit files, okay, and you open evolutionary theory, and you open the first topic, which is the theory of evolution, whatever. I have to go to the evolution unit and open evolutionary theory. there is a folder there that's basically a download of a YouTube channel. I got every single video from their channel. I'm going to show you that to you. So this is again on the videos and tutorials folder, not the podcast folder, videos and tutorial folder. There it is. Think about it. All right? Now, there's two types of videos here. The first video series that they have is pro-evolution. So this is just evidence describing what evolution is, how it works. This will truly show you what evolution is. Now, before someone talks against evolution, they should at least understand what it is. So that will give you a good job in understanding the logic and evidence behind evolution. It's my favorite YouTube channel on evolution. But the guy is also an atheist, which I'm not, but I'm saying, so, but why did I put the anti-creationism folder there? For one reason only. Not because I'm actually against religion, but because I'm against stupid religion arguments, right? So I don't like when people say things that to defend their faith with empty logic errors, right? Because that makes you look foolish. And it makes us lose our integrity, so to speak. So anti-creationism, I like this folder because it's about, hold on, because it's about debunking bad arguments that they make against evolution. You follow what I'm trying to say? So check it out. Next class, please remind me, I'll start with the video of homologous gene evolution, which is very important, chapter 21. I'll see you guys next class. Actually, no, next class is planting day, the class after that. Make sure you know how to plant the plants. We start next time.